Ah, I'm eating my apple, so I was not ready. Okay, we're gonna hit gel electrophoresis on this one. Well, okay, so cool things about gel electrophoresis. We can use this for lots of things, but the premise of it is that DNA is slightly negative, and because DNA is slightly negative, we can run a current through it, and it will run through an agarose gel. Okay, what this will do is it'll kind of look like this. If you put it under a black light, you'll see these bands, and each band represents a fragment of DNA, and it separates those DNA fragments based off of their size. So if I am a small piece of DNA, I'm going to run really, really far away from these things called wells, which is where I initially load them. And if I'm a fat piece of DNA, I'm going to stay home really close to the wells. Now, important thing to know, and I think on your test, this is actually something that uses to separate nucleic acids, but it can also separate proteins. And then I taught you guys that the bands can look like they're on top of each other if they're the same size. And so just be particular and make sure you remember that, if they're going to be the same size band or the same protein chain, because you can do polypeptide chains based off of this as well, that same size will run to about the same place. So that's why some of the bands maybe look thicker, is because there are a couple bands stacked on top of each other. Okay. So one of the things that we use this for is crime scene analysis. Like, other than the fact that this one is highlighted and so is this one, we can tell that this sample of DNA taken from the crime scene is matching suspect 2 because of these DNA fingerprints. Now, important thing about creating DNA fingerprints is it is based off of the idea that every single person's genome is different sequence of genetic code. So it's like A, T, C, G, G, A, C, D, no D, D is not a nucleic acid, but you know what I mean? It's just a bunch of series. And we use the same restriction enzyme to cut at different lengths. And so we've got areas where they have fragments that are really small, are really large and don't go travel very far, versus ones that are very small. And everybody's going to have different sized fragments based off of that. And then we can also do paternity testing. One thing to keep in mind with paternity testing, you can compare the mother and the child, and you can see here's kid's DNA. They got this from their mom. This DNA is from kid, so it could be from either parent 1 or 2, potential father 1 or 2. Here, it's something they got from mom. This double band right here, they clearly got from dad 1. So we can eliminate dad 2 as well. And so father 1 is, our person 1 is definitely the father. And that's how we do it for paternity testing. Sometimes in kids, these rogue bands show up that it's like, where the heck did this band come from? That's because of crossing over. So we're not getting a identical banding directly from a mom or a dad. It's like that combination of genetic code from the mom and dad. All right. So Mo's Guide to Gel Electrophoresis. 1, PCR is the first step of any gel electrophoresis. We've got to do a polymerase chain reaction. So we actually know what polymerase means. Polymerase is that pairing of nucleotides. And so what this does is it amplifies, is the word they're going to use, but we also use it to make copies of DNA. So instead of just one double strand, I've got lots of copies of that same DNA sequence, so that my bands show up thicker in the gel. It'll also repair any, say we took a sample from a crime scene and we're missing part of the double strand because it's been damaged. We can easily repair it based off of complementary base pairing. Basically, PCR is a polymerase with a bunch of free floating nucleotides so that it can repair. Restriction enzymes, super, super important in this process. What we're going to do is we're going to cut, or cleave is the fancy word, the DNA into these different sized fragments. Then we're going to run the gel. We're going to take the negative DNA, load it into the wells, and then run it through the agarose towards the positive charge. And then we will analyze it based off the smaller fragments traveling faster than the larger fragments. So they will have traveled longer distances in the thing. All right, so we can also use this for like genetic testing or screening or using restriction analysis. So we can actually cut out just a sequence of a genome where we're looking at a gene. So for instance, we're going to look at the sickle cell gene, and we can compare the sickle cell gene to a normal allele by cutting out just that gene. So we can really kind of dive into understanding like genetic disorders and how they are slightly different than the other. And remember, this can be in proteins or it can be in nucleic acids. And another really cool thing about gel electrophoresis, so it's like, why do we care? What's the deal with gel electrophoresis? Well, it's really important for us to understand a map of a plasmid. Because if we understand a map of a plasmid, which is bacterial DNA, then we can take that plasmid, cut it open, and insert a desired genome so that it produces the correct thing for us. So when all is said and done and we look at a plasmid map, we're going to see different bands. We're going to see different band lengths or different fragment lengths all the way across here, and different enzymes that can cut that. And so we use different enzymes so that we, get a lot of variation between, and we can actually create this map. And again, why do we care about this? Again, it's so that we can insert desired genes, have an understanding of what the genome looks like of a bacteria, so that it's easy for us to manufacture pharmaceuticals. So again, this is the why. If we understand a plasmid and bacterial DNA, what we can do is we can take that gene of interest, insert it, and then this guy's going to do all our dirty work for us. He's going to reproduce. When he reproduces, he's going to make even more bacteria, creating proteins from the desired gene that we want. And it can be things for, we have some bacteria that is making us bacteria that cleans toxic waste. We have some that are pest resistant. We have some over here that are creating human growth hormone for us, or insulin, or any of that crazy stuff. So let's map it. I just spent a ridiculous amount of time trying to find a pin. I found the pin. Okay, so we're gonna map this bad boy. This enzyme X, pow, see, found it. Not very good at drawing, turns out. Is going to cut this plasmid and make two fragment sizes. The sizes are 7.5 kilobase pairs and 2.5 kilobase pairs. So that means this whole thing is 10. Okay, kilobase pairs. Now, when we do this, I like to start by looking at a single enzyme cutting the most amount of time, because I can put that one on there and then I can figure it out from there. So like I've got this one to here this is going to be 7 point. Yeah, this is great. I love this for me. This is the 7.5. Loop. And then that means this is the 2.5 from here to here. All right, now that I have done that, I have to figure out where enzyme Y is going to go. And so enzyme Y only cuts one time because that's the total thing. If you look, this added up to 10, this with one cut adds up to 10, so that's where we get our total from. So I have to put this cut on there somewhere. Well, if I look, this 2.5, okay, and this 2.5 are the same. So the cut does not disrupt that fragment. And remember, enzymes cannot cut. This cut cannot be where an enzyme already is. So I've got to cut somewhere on this line to make a boop, a 1.3, 1.3, and a 6.2. Yikes. And a 2.5 is there from earlier. Okay, so that's kind of how you do that. And then this thing is you're just doing it. So remember this first one, this is if there were 10 base pairs. I almost messed up, but I fixed myself. This is if there were 0 base pairs. The X enzyme, there's going to be a 7.5 and a 2.5. The Y enzyme is going to be a 10. Okay, from the well, it pretty much stayed in the well, it's so fat. And then this one is going to be a 6, a 2, and a 1. Okay, and that's kind of how you do that. All right, exit that drawing. Cool. And now I've got this laser pointer turned on, and I'm not really sure why. But you could have put those Y cuts here. You could have put it here, as long as it's in the correct place. All right, so that was Mo's guide to gel electrophoresis. Hopefully that one helped. It's pretty easy. It's lots of application questions. And moving on to the next one. Yay!